Hey everyone, how are you all doing? Hope good. So, welcome back to my channel Priyanka's Education Videos, and today's topic is the a continuation of last topic, which was about bacterial nutrition. And today, in this video, we shall see about bacterial reproduction. So, let's get started. So, bacterial cells reproduce in two ways: asexual reproduction and sexual reproduction. So, asexual reproduction takes place by binary fission, conidia formation, budding, cysts, and endospores. So, let us start with binary fission. Sorry, before that, we'll see the ways of sexual reproduction that takes place through transformation, transduction, and conjugation. Okay. So let's start with the asexual part, that is by binary fission. Now binary fission is a kind of reproduction in which a single bacterial cell divides into two daughter cells. So in the very beginning of the process, when the cell is completely grown and is ready to divide, initially, so the first step is the genetic material or the nucleoid of the cell, that is nothing but again the DNA, undergoes replication, and the two strands of DNA are formed, and the nucleoid of the cell is divided into two parts, that is two newly formed strands of DNA are formed, and these uh, formed strands of DNA move towards the poles of the cells, that is the corners of the cells. Then the cell slowly starts to elongate, and a cross fall or a septum is formed in between the cellular components. Now, after the formation of the cellular components, the two new daughter cells get divided or get separated from each other, thus dividing the parent bacterial cell into two daughter cell. So let us see how does this happen through a diagrammatic representation. So this is the diagrammatical representation of how exactly the asexual reproduction through binary fission is taking place in a bacterial cell. You can see in the very first picture, that is the first step, where you can see the normal structures, which are cell wall, cell membrane, and you can see an elongated nucleoid. Now in the second step, the elongated nucleoid is dividing into two and is getting separated from each other, and it is moving towards the poles or moving towards the corners of the cell, and the cell wall and the membrane are protruding inwards, forming a cross wall or a transfer septum. Then later on in the third step, you can see uh, the separation of two, or the transfer septum is completely formed, dividing the cell into two equal parts, and eventually the daughter cells get separated. So from each bacterial cell, two new daughter cells are formed and they get separated. So this is how the binary fission, a kind of asexual reproduction, takes place in the bacterial cell. Now next is conidia formation. So this is one more type of reproduction that is taking place through the formation of conidia, and is this conidia is found, or this kind of reproduction is found in filamentous bacteria such as streptomyces. So the part of the filament that bears conidia is called conidiophore. The formation of conidia takes place at the apex of the filament through the formation of a septum. The conidia are small, spherical, chain like or spore like structures. Now, after the formation of these conidia, they get detached from the filament, and upon reaching the suitable substratum, they again they germinate and again grow into a new mycelium. So let us see. This type of asexual reproduction is also called as fragmentation. Okay. Now let us see the diagrammatic representation. Now you can see the filamentous bacteria, how it looks like, and you can also see in this diagram or in the picture the formation of conidia at the apex of the filament. So at that particular point, the conidia or the conidium is formed, and slowly they are, or the below structure which is bearing the conidia is called as conidiophore. And you can see the after the full formation of the conidia, they get separated from the conidiophore, and they get dispersed, and upon reaching the suitable substratum, they again germinate into the mycelium, which again will form into the new bacterium. So this is how the asexual reproduction through the conidia formation takes place. Now next is through budding. Budding is almost similar to the binary fission. It is also a type of asexual reproduction in which bacterial cell develops a small outgrowth or a bud due to cell division at that particular site. These buds develop into tiny individuals. Simultaneously, nucleus also undergo division, and single nucleus with some cytoplasm enters the bud. After the bud gets matured, it gets detached from the parent cell. Now this is how the process of budding looks like. So a bud begins to form from the parent cell. Later on, at that particular point where the bud is formed, you can see a small the cell is protruding outwards. Now after that, the nucleus starts to divide, or the nucleoid material is copying itself and is dividing into two or forming the new copies, and the bud also is slowly receiving the nucleus and also the cytoplasm. Now the newly formed bud becomes a separate daughter cell and eventually gets separated from the parent cell. So this process of budding produces chains of cells. Next is through the formation of cysts. So these are the dormant or resting structures of the mother bacterial cell, formed by the deposition of an additional layer around the cell wall. In these conditions, the metabolic processes of the bacterial cell are slowed down. Under favorable condition, these cysts wall break through the process of excistation and germinate to form a new bacterium. The main function of the cyst is to protect from the adverse condition. Example, azotobacter. Now this is an example or the representation of the formation of cyst in the bacteria. Next we shall see the endospore. Endospore are also the tough structures like that of cyst, which are formed inside the vegetative cell. So these are tough, perennial, dormant, resistant, and specialized structures formed to combat unfavorable conditions. The endospore producing mother cell is called sporangium. Only one endospore is formed in one bacterial cell, and on germination, it gives rise to a new bacterial cell. The bacteria can withstand exceedingly high and low temperatures, acidic and basic conditions, and large amounts of radiations in the form of endospore. The endospore has many wall layers containing heat resistant chemicals such as sialic acid and dipicolonic acid. So you can remember these important points, uh, which are very important objectives. So these can be easily dispersed through wind, water, and the gut of animals. On the onset of favorable conditions like that of cysts, endospore break to release the bacterial cell. Example, bacillus and clostridium species. Now uh, this is the diagrammatical representation of the spore or endospore formation, which is including several stages. And in the final stage, there is the complete formation of cyst. You can see, yes. Then upon reaching the favorable conditions, the endospores are again turning back to the normal bacterial cells. So this is all about the reproduction that is taking place asexually in the bacterial cell, that is through binary fission, conidia formation, budding, cysts, and endospore. I hope the video is clear, and the later on the next video, in the coming video, I shall be explaining about reproduction which is taking place sexually, that is sexual reproduction in the bacterial cell, that involves the exchange of genetic material. So I hope this video is clear. Thank you for watching.